When you look at biochemistry as a field, it's almost always taught as here's all the different parts. You've got your proteins, you've got your DNA or your genetic information, and then you have all the enzymes and all those biochemical pathways. So, okay, well, what is the first enzyme in glycolysis? What is the second enzyme in glycolysis? What are the different pieces that you need for glycolysis to work? How do those structures change? Okay, or they become different things. And it becomes a lot of naming and individual pieces. Okay, so I looked at that and said, well, that's kind of weird. Okay, and I had an interesting experience as an undergrad because my degree is biochemistry from a chemistry department. And my girlfriend at the time was biochemistry also from the biology department. We both took a biochemistry class at the exact same time, but the material taught was entirely different. Her process was, what is the name of this molecule? What is the name of this molecule? What is the name of this molecule? What is the name of the next thing, and the next thing, and the next thing? Where I was taught, this is the structure. We don't really care what the name is, but it's this. What happens to this structure? Where's the most reactive site within it? And how do you break that down? Okay? And you can kind of follow through the rules of organic chemistry how things degrade and how things should be built up. And you can apply a lot more knowledge to what's actually happening within it. Okay? What does a biochemist refer to when they look at where biochemistry comes from? 9 times out of 10, they look at it and they say it comes from biology. Okay? And they're looking at this larger overview of what's happening. Okay? They understand very little about what's happening to each of the individual structures. As an optimist, I would say it's actually coming from bunch of functional groups, ultimately organic chemistry. But again, ask any biochemist how much organic chemistry is present, and they will tell you 0, which is mind-bogglingly ignorant and stupid as far as I can tell, because everything that is happening in biochemistry is organic chemistry. So those of you that loved organic chemistry, it's going to come back again and again and again. Everything that's happening within those is how do those structures interact? What are they interacting with? What are the most likely reactions? What are the intermolecular forces allowing these things to touch each other? Okay? It all comes back to organic chemistry. You are looking at those functional groups and how those functional groups can build larger molecules. Okay? So you need to be familiar with the general functional groups. Your textbook has a nice table of them. I put out a summary of some other ones. Some of these, well, let's kind of go through them. Let's see what we got here. Let's push at your chemistry knowledge. Anybody recognize any of those? Benzene. Okay. And we're actually going to change that. Benzene's not going to be the best name. Okay. That structure is technically benzene, so fair enough. But that's not your best answer in biochem. Okay? If that is indeed benzene, what we were saying is there are hydrogens attached to all of these. Okay? Which means, what can this structure then connect to? Rhymes with something. It can't bind to anything. It's nothing. Because it's not cyclohexane, it's benzene. Okay? There is no bond location. It can't build structures out further because carbon can only have 4 bonds. And each of those points, being a carbon, already has 4 bonds. So when we're looking at this structure, we aren't referring to it as benzene. We're referring to it minimally where it has some place that it can connect. We'll just write it as connect there. In which case it's not benzene anymore, it's this will keep coming up. Phenyl, not phenol. I know that's irritating. We will see that again. We're looking at a phenyl group with a YL. Okay, what were some other functional groups in there? We've got the aldehyde. We have a ketone. Where's the ester? Sorry, no ester. We have an ether. The far right is carboxylic acid. I guess I kind of gave that away as I wrote C. Arguably, the carboxylic acid is one of the most important functional groups, okay, for biochem. What's the other important functional group for biochem? The amine, and as you guys are shouting things out, you'll notice that we have different pronunciations. It's not a big deal with the pronunciations. In most cases, there's not enough overlap that I'll understand what you're trying to say. The phenyl one is tricky because you get phenyl or phenol. Okay, amine, amine, but not imine. Okay, with an I, so we got to be a little bit careful with those. Your amines and your carboxylic acids are arguably the most important. Why are those the most important?
Acid bases.
They are acids and bases, yeah. What is special about the nitrogen in the amine? Amino what? Amino acids. You mean amine and acids? Proteins are amino acids. Amine acids. Okay. That's why they're critical importance in functional groups, because they are proteins. That's what gives us proteins, is the amine functional group and the acid functional group. Okay. Another big one that becomes important would be the ester, which is ultimately just taking a carboxylic acid and tweaking it slightly, that last oxygen is connected to something else. Okay. A carbon or a phosphorus, something else. Okay. We're looking at an ester functional group. Oops. Okay. The other ones do pop up, and we will have to look at their intermolecular forces and how they cause major interactions. But for the most part, our bonding structures are really settled in your amines, your acids, and your esters. So we end up with a couple big themes. We've already kind of vaguely mentioned some of these. Lower right-hand corner, what do we have? ATP synthase, which we would classify as what category for biochem? We could go with enzyme. We can go a little bit broader, though, as a category. We're looking at a protein. Enzymes can be proteins. They could also be RNA. So enzymes is a broader class that goes over larger aspects. What do we have on the far left? We're looking at DNA. So when we're looking at our broad themes for biochem, we can usually drop it down into our classifications of proteins and DNA. Okay. So our genetic material and our kind of active material. So we're looking at the same biomolecules. Those biomolecules interact through organic chemistry, and they're all going to end up using energy, which comes from physical chemistry. Okay. The physical chemistry ties that we're looking at are largely centered in 152. Okay. How many people liked 152? Wow, you liked it?
Yeah, that was all right.
Oh, cool. Who'd you have as an instructor?
Giletti over
Okay. Yeah. I'm just curious. So yeah, we're going to use a lot, or not a lot, but a sizable amount of kind of physical chemistry aspects that are usually centered pretty hardcore on 152. Okay. So we're going to look at thermodynamics and kinetics, and we'll look a little bit at some kind of acid-base titrations that you'd be used to. Okay. And then all biochem typically starts off with this weird kind of philosophy. Biochemistry as a field is kind of closely tied to religion, and then what we were looking for is a why. How did life start? What is life? So you get a big philosophy spin on it. Okay, so from the inanimate to the animate, what did we start with? Any ideas? What atoms? Did we just have individual atoms floating around, or did we have small molecules that started to build things? both cheap. You got the slides too. Okay. We did ultimately start with just free atoms, but those atoms end up being very reactive and start to build larger molecules. Those larger molecules end up giving us the small molecules that we potentially, arguably, started with. Okay. So when we look at our early Earth environment, there are still, of course, up for debate, a sequence of small molecules that are potentially available to us. Okay, and this is some of them. You've got ammonia, you've got hydrogen sulfide, carbon monoxide, carbon dioxide, methane, nitrogen, hydrogen, and H2O. So from these basic building blocks, somehow or another, we scaled up to the massive systems that we have today, like DNA and ATP synthase. Okay. If you want to get an idea on size, those molecules, largest one there is carbon dioxide. Yeah, carbon dioxide with a mass of what? What is the mass of carbon dioxide? Single molecule of carbon dioxide. 46, let's just say round to 50, make sure we covered it. Okay, let's even be nice, let's round to 100. 100 grams per mole. So one molecule of this, roughly 100 grams per mole, or 100 AMUs. If we go up and now look at the size for ATP synthase. What is the mass of that thing? 355,000 grams per mole. It's a couple orders of magnitude bigger than CO2. Okay. That's kind of impressive. How can we go from something that is 100 grams per mole to 355,000 grams per mole? This is a pretty massive scale up. How could that possibly ever occur? And this is kind of your philosophy, and this is something that biochemists, or at least some biochemists, are still working on, but it's kind of pushed to the back burner because it's very hard to prove any of that. With all of these gases, some of those are up for debate. Well, we don't think we had liquid water, so you can't have liquid water in your environment. Okay, well, if you remove that, then your experiment changes. Okay. Miller and Urier took a whole bunch of these, and they mixed them in a container, and then they threw in a bunch of other pieces. Okay? One of the other big pieces that we're going to need is that if we just take those molecules and sit them near each other, not a whole lot's going to happen. Okay? Ever. Okay? Because for them to actually do something, they have to have energy. Well, then we go, okay, well, what energy sources might we have had at early Earth? Now we run into yet another debatable issue. Well, should we have volcanoes? Should we have sunlight? Okay, what are the possible things that we have access to? So Miller and Urier just went through and decided what they had. They built an experiment, an entirely closed system. Believe it or not, it is still running as an experiment. Kind of neat. Every so often, they pull something out to test it, to see what's going on within it. But it's a closed system, and what they were trying to do is say, if we just take all these random things, what could possibly get built out of this? Okay. They started it, anybody remember? I'm going to say the 40s. I don't remember. But what they were able to find is they ended up with a couple small molecules. We had hydrogen cyanide, we had an aldehyde, formaldehyde, and some amino acids. Why is this really important? Well, if we just take a bunch of random stuff and we allow random action to occur, what do we get? We sort of get the foundations or the beginnings of the pieces that come together to eventually make life. Okay? That we don't need some outside external control, that it can happen through random interactions and random motions. Of course, this becomes a challenging experiment to deal with because, number one, everybody's then going to nitpick everything you've put in here. Okay? Number two, they'll also nitpick, did you make sure that every single piece of your apparatus was entirely closed to the rest of the outside world? Okay? That was a fun discovery that I'm not sure if it happened with the moon, but it might have been from the moon. They took some rocks back from the moon. They're, oh my gosh, we found life on the moon. What happened? They took life up with them, and then they contaminated the moon rocks and brought the moon rocks back and said, oh, look, we had life. Whoops. You got to be careful with your system. Okay? So a lot of those kind of arguments go back to this and saying, well, it was contaminated with something else to begin with. Okay? There is enough evidence to suggest it wasn't contaminated, that they actually did a pretty cool experiment, and that we are getting small molecules coming out of that. Cyanide and formaldehyde are big pieces, or big important molecules to include, even though they are relatively small. Why would they be important to include? What is special about those two molecules? Maybe it would help if we looked at a Lewis structure. This species is a weak acid that would eventually turn into H+ and Cn-. What is special about Cn-?
Pretty reactive.
How?
Can bond with another CN. You can
It can't bond with another Cn.
another C. And add a carbon.
How? What is special about this being negatively charged? This species can act as okay, so, and this is going to come from different backgrounds, a base. So in this case, if we looked at it just acting as a standard Bronsted-Lowry base, that's pretty boring. So by base, what do you mean? Bronsted-Lowry? Or would be a Lewis base, so you're looking at it acting as a nucleophile. So this is something that when I teach organic, that I'm very kind of nitpicky on. The statement base, while true, that's boring chemistry if we look at just the Bronsted-Lowry definition. So when you're referring to it acting as a base, you're actually referring to it as a Lewis base, okay, where it's sharing electrons with something else other than hydrogen. Okay. To clarify that, I look at acids and bases solely as Bronsted-Lowry, transfer of hydrogens. And I look at the transfer of electrons to something other than hydrogen as nucleophile or electrophile. Okay. And we will come back to those definitions again. What's special about formaldehyde? No ideas? If we go back and take a look at this guy, why might this guy be acidic? Why would that carbon take electrons from the hydrogen? I like looking at the periodic table.
Just thinking of the electronegativity of nitrogen.
Nitrogen, being more electronegative than carbon, starts to suck the electrons away from the carbon. Carbon then starts to lack electrons. So what is it going to try and do? It's going to need to get electrons. The only place it can get them from now is from the hydrogen. So it takes electrons from the hydrogen, we lose H+, and our carbon now has that negative charge. By looking at the electronegativities, we can predict that we would end up with Cn-, which would give us our nucleophile. Use that same thought process over here. Oxygen is very electronegative. It is the second most electronegative element on the periodic table, which means what does it do to these electrons? Sucks them away, which then means the carbon could try and take electrons from the hydrogen. We run into an issue of it not being able to stabilize that negative charge well. So it can't take electrons from the hydrogen. Instead, what happens to the carbon? Becomes positively charged. So this negative charge could come in and share electrons, causing that bond to break. This is a nucleophile. This reacts with nucleophiles. What reacts with nucleophiles? Electrophiles. So these two molecules are of critical importance because we can now potentially use these to build our amino acids. Okay? When we get into looking at amino acids, remember the distance between your carbonyl compound, or your oxygen-containing carbon, and your nitrogen-containing carbon. Okay? And apply those back to looking at those two structures. Okay? It should look frighteningly familiar. Okay? Within all of this experiment, kind of neat because they went through and they said, oh, proteins exist. Okay? Within that context, we now say, well, if proteins were the start of life, they must be the ones that carry genetic information. They must be the ones that then build life to a larger scale. Okay? And as neat as this experiment may be, they've gone through and changed their minds about the origins of life, and now it sounds like RNA is more likely to be the origin of life, not proteins. Any ideas why? What do you remember from biology? Okay, why would RNA come before proteins? Okay, I think I know what you're referencing. If we looked at DNA, it goes to RNA, that then goes to proteins. Okay, we're going to look at synthesis. So that's already looking at a built system. We don't have a built system here. So why would we expect RNA before proteins? Because if you make that argument, then you say DNA should start, not RNA. What is the role of DNA? Genetic information. Okay, that is it. Doesn't do much of anything else. We look at our proteins. What do our proteins do? Proteins give us structure. They give us enzymes. They can generate different reactions. What does RNA do? It makes the proteins. But to make the proteins, what does it need to contain? Genetic information. What does RNA have? It has both the aspects of DNA and proteins. It's kind of that intermediary ground. Okay? Why would we not want something that just does everything? What happens if that everything fails? You're kind of screwed. Okay? So we could start with RNA that then branches off and says, okay, I need something that's more stable so that when it fails, or it's harder to fail, okay, that can be shifting to ultimately a hard drive like DNA. Okay? RNA that, if something else failed, could potentially do some enzyme catalysis and do reactions. It has been proven that that can happen. Okay? But it's not very good at it. It doesn't have the same flexibility that, say, proteins do. So what should we potentially evolve? Proteins that can now be a little bit better at that job. RNA can kind of act as this intermediary to kind of balance out both of those. Okay? Kind of a neat little process that we're going through. So that's one of the reasons why we tend to now think that RNA is potentially our origin molecule. How do we move from this philosophy to chemistry? Okay. Now we do this big, massive jump, because in the philosophy it's, well, how did life start? What are all these things? Okay, now we're going to end up jumping straight ahead and saying, okay, I don't really care too much about how it started, but now that I have these pieces, what can I do with these pieces? Okay. Those pieces are our amino acids. The amino acids can be used and put together to generate polymers. Okay. And in organic chemistry, sometimes, depending on your instructor, you may have heard reference to polymers. Sometimes they go into it more detail than others. Okay? What are proteins? Polymers. But we never call them polymers. What do we call them? Polypeptides. Why? Because a biochemist a long time ago looked at organic chemistry and said, I suck at organic chemistry. I don't want any reference to organic chemistry because I want my future students to think that organic chemistry has nothing to do with biochem. So we're going to call it polypeptides. You can see my opinion starting to seep through. Okay? They are polymers. Okay? So that can get us to our polypeptides, our proteins. We can take sugars. Okay? Those of you who remember chair configurations, okay? Fisher projections from organic. Guess what? Comes back with a mighty, mighty huge vengeance with your sugars and your carbohydrate chemistry. Those sugars can come together to make, okay, carbohydrates or polysaccharides, or for Christ's sake, a freaking polymer. It's the same thing. We're just using a different repeating unit. And by using that different repeating unit, we're still building a polymer. We just have to name it something else because biochemists don't know how to name things. Okay? If we go through and look at the individual pieces that build up our genetic information, they're nucleotides. Those nucleotides, when polymerized with each other, form nucleic acids. So we're taking these individual small building blocks and we build them into larger and larger pieces. Depending at what level we want to look at, we may want to know something about the structural detail happening all the way down at the bottom. Okay? Because that structural detail does affect the top. How much depends on what you're trying to study. Okay? Oh, my favorite slide. Everything about biology in one slide that I could care less about. I covered the money, hopefully. So as we continue to build these things out, we start to separate and build compartments that can build larger and larger complexities so that if one system fails, other systems can take over. Okay? We don't want one thing controlling everything because then ultimately the whole system crashes. We look at two primary categories, your prokaryotes and your eukaryotes. Okay? We have these two primary categories because they do things differently in each system, which then means when you move to DNA, okay, replication, what are the other ones? Transcription, translation, all that fun stuff. Depending on the complexity of your organism, you're going to get slightly different rule sets or slightly different molecules involved in it. So when we and or you talk about DNA, you're going to be referencing prokaryotic DNA synthesis or eukaryotic DNA synthesis. Which one do you think we know more about? What? Which one do we know more about?
I think prokaryotes, they're easier. I don't know.
Why are they easier?
They're smaller.
Okay, smaller, what else?
The bacteria, they grow, they replicate faster.
No nucleus, no mitochondria, no endoplasmic reticulum. They are vastly more simple. Okay? They can grow fast. We've got a lot of those aspects that make it easier to study those molecules, those cells over our eukaryotes. Okay? I always get them backwards too. Okay? So feel free to point that out. Okay? So you're kind of responsible for recognizing which one's more complex than the other. To a lesser extent, you should know some of the details behind it. I'm not too concerned about it. You might see one multiple choice question on that. You can then look at classification. Lovely picture. You've got all the, what are they, kingdoms? Actually, here, let's do this. How many of you know the kingdoms?
Like 3 different classification systems now or something.
I have no idea, actually. It's all biology to me. I was just curious. So what do we get? Fungi, plants, animals, Protista, Monera. There's some fun biology themes that start up in that first chapter. Don't stress on them. You should read it, be somewhat familiar with it. I don't think I would ever test on this. Okay. One of the more important things to look at would probably be back to our chemistry aspect. This is arguably one of the most important biochem reactions. Why? What do you mean energy?
It's how we move. It's our energy source.
ATP is our energy source. I agree. This reaction is of critical importance for transferring energy. But you look at this equation, where do you see energy?
There. Isn't it released?
It's a good question. Why is it not in the equation? Biologists.
Root of all evil.
There is an energy release within this reaction. Okay. That energy can be transferred or consumed by other reactions and allow this thing to process. Okay. This is where our energy source is coming from. Okay. How could we potentially harvest or manipulate this? Well, to do that, we'd have to understand something about energy, which we'll pick up on the next day, Thursday. Real quick, second question. What type of energy transfer is this? I don't know, it's kind of a weird question. Fine, way to answer the question. Exothermic. Okay, as vague as it was, you're too used to my vague questions. It's an exothermic reaction. We've released energy. If we turn the reaction around and ran the reverse, it would be endothermic. So it's important to reference those. We'll pick up there on Thursday.